करें?
Radium.
और कोई?
Inculcation of plant growth promoting microbes that improve the nutrient capacity of the soil and increase the nutrient uptake of the plants to promote their growth. Biofertilizers.
Yes, yes. और कोई बता सकता है कुछ?
Living microbe that enhance plant nutrition by either by mobilizing or increasing nutrient availability in the soil.
Okay. Okay, so in the last class, we have discussed about the biofertilizers, in which we have discussed about the biofertilizers, and we have told that biofertilizers are fertilizer with life, and life means the millions and millions and microbes are associated with the soil also, and in the case of fertilizers, carrier materials carries these microbes in huge quantity in sufficient number, and efficient microbes are mainly supplied to the through the biofertilizers, and they are capable of fixing nitrogen or supplying or mobilizing phosphorus and other nutrient and other plant growth promoting substances in the rhizosphere, and that are helpful for the plant growth. They are able to increase the availability of nutrients by the different processes, and they improve the plant growth by the supply of nutrients. Okay, so there are several biofertilizers. We have discussed also about this that, and the most important one is the rhizobium, which one is the most important and widely used also. They are exploited in every sectors. Okay, so rhizobium is the most important, and for the legume crop, they are important as they supply nitrogen by fixing atmospheric nitrogen in their roots by making a root nodules in the plant roots, and in that root nodule, that is the site of nitrogen fixation that contain leg hemoglobins, and the microbes rhizobia are imbibed in the leg hemoglobins and fix atmospheric nitrogens. And after fixing atmospheric nitrogen, they are converted into the ammonia, and they are converted into different amino acids and transferred to the plant systems for the conversion into the proteins. Okay, so rhizobium is the most important, and they are mainly associated with the legume crops. And any other crops which are important for the rhizobium, where the rhizobium inoculation should be there? बोलो जी। पल्स तो लेग्यूम ही होगा, इसके अलावा और किस किस क्रॉप में राइजोबियम इनोकुलेशंस होते हैं? कुछ वाइल्ड सीड्स बताओ। सोयाबीन एंड ग्राउंडनट वाइल्ड सीड्स में आते हैं, ठीक है। इसके अलावा फोडर क्रॉप्स में, in fodder crops of barseams, so in the barseam there is a root nodulation also. Spices में बताओ। Spices में मेथी है ना, so methi also have root nodulations in the same way. And the another important crops is the green mandarin crops. Several green mandarin crops have root nodulations with the rhizobium, and the most important one is the sesbania. Okay, so in the root nodulations, legume rhizobium important. Non-legume rhizobium में आपका आता है Pasponia, Frankia, Elnas, Marica, where the there is a nodulations will be takes place with the help of actinomycete, that is the Frankia. Okay. Another one is the stem nodulations is also takes place in the sesbania. In case of sesbania, stem nodulations takes place, and nitrogen fixation also takes place in the stems, and so the nitrogen fixation in the stem is also called philotrophic nitrogen fixation. Okay, तो सिस्वेनिया में ऐसा है, इसमें दोनों नोडुलेशंस होते हैं, भी होते हैं, स्टेम नोडुलेशंस भी होते हैं, है ना, but it depend upon the conditions. In a stem nodulations, they are mainly takes place in Sesvenia rostrata. Okay, so it is mainly in the deep rice fields or where the waterlogging situation is there. In that conditions, nodules are formed in the stems. But when there is no waterlogging condition is there, the nodule will be formed in the roots. Okay, so in the case of Sesvenia, nitrogen fixation takes place through the rhizobium. Another one is the Casuarina, in which the stem nodulation takes place with the Frankia. Frankia is a group of actinomycetes that fix nitrogen. Okay. Another non-nodules forming nitrogen fixing process are in Azolla fern. So in Azolla, blue-green algae is associated with these Azolla ferns, and that fix atmospheric nitrogens. Another one is the moss helosiphone is also where the nitrogen fixation will be takes place. Another non-symbiotic process. So earlier we are discussing about the symbiotic process, in which there are symbiotic association with the rhizobium and plant roots are there. They live in the plant roots, and nitrogen fixation takes place. Okay, so some non-symbiotic processes are there, like Azotobacter. Azotobacter is free living. They freely live in the rhizosphere, and through the multiplication they increase their populations in the rhizosphere and fix atmospheric nitrogen. Although their fixation capacity is very low, but these Azoospermia, Acetobacter, Bijrinkia, Clostridium, Cyanobacteria, Anavena, Nostoc, Dersia, all these are able to fix atmospheric nitrogen. From 15 to 40 kg of nitrogen fixation will be there. Okay, so these are the non-symbiotic nitrogen fixation. There is no association with the plants, roots, or any other things. They live freely into the rhizosphere on the water body also, like in the case of cyanobacteria, Anavena, Nostoc, Dersia. Then the phosphate solubilizing, like the PSP. PSP means phosphate solubilizing bacteria, and PSF, that is phosphate solubilizing fungus, and PSM, that is phosphate solubilizing microorganisms. Okay, so phosphate solubilizing bacteria, several bacteria are there. Two are important, like Bacillus polynesia, Pseudomonas ostreata is there. Another one is in the case of fungus, Trichoderma is there, Fusarium, Aspergillus, Penicillium. These are of mobilizing phosphorus into the rhizosphere. Another one is the phosphorus mobilizing, that is VAM, vesicular or vesicular mycorrhiza. So fungus, these are fungus mainly. In case of glomus, gigaspora, these are the example of the phosphate mobilizing capacity of the vesicular or vesicular mycorrhiza. Another one is the potassium solubilizing. क्या
some students join sir, कुछ students join करना चाहते हैं sir.
ओके, प्रिया भारती केवल है. एक ही स्टूडेंट था जी.
सर दिव्यांशु मिश्रा भी था.
और तो कोई आ नहीं रहा है अभी. उसको कौन ज्वाइन करने के लिए जल्दी.
ओके, सो हो गया सस्पेंड बंद।
ठीक है। So we are talking about phosphate solubilizing bacteria, phosphate solubilizing microorganisms. So if PSV is there, then phosphate solubilizing bacteria like Bacillus polymedia, Pseudomonas ostiata, Pseudomonas putida is also there. Even the for phosphate solubilizing fungus, Trichoderma, Fusarium, Aspergillus, Penicillium is there. Phosphate monobilizing, that is vesicular glomus is there, Gigaspora is there. So all these microbes are comes under the PSM, phosphate solubilizing microorganisms. Then potassium solubilizing, that is the Fracturia aurantia bacteria is there. Even some bacillus are also useful in the potassium solubilizing capacity. Then organic matter decomposers are also there, like cellulitic and lignolytic bacteria. Cellulitic, that is Trichoras, Trichoderma, Chaetomium, then Bacillomyces, Cytophaga are there for the cellulotic microorganisms which decompose cellulose. Then lignolytic are Pleurotus, Agaricus, Polyporus forms are there. So these are the classifications of the different biofertilizers and their microbes which are associated with the different type of functions like nitrogen fixation, phosphate solubilizing capacity, and mobilizing capacity, potassium solubilizing capacity, and organic for the organic matter decomposition. Then there are different type of nitrogen fixer also, like we have discussed all these things like free living. In case of aerobic, they are Azotobacter, Bismuthia, Klebsiella, Cyanobacteria. They are aerobic, so they require less amount of water. They remain on the surface like Cyanobacteria on the surface. Azotobacter in the rhizosphere, but they the water conditions. In the anaerobic condition, Clostridium, purple sulfur bacteria, green sulfur bacteria, they are anaerobic. So in the deep water, they can found and they can do the function of the nitrogen fixation and other sulfur solubilizations also, oxidations also. Symbiosis with plants, in that case, legumes are most important where the symbiotic nitrogen fixation takes place. These are the classification of rhizobium. Azorhizobium, Elorhizobium, Mesorhizobium, Cynorhizobium is there. Other plants like Azoospermum, which is micro-associative. They are associated with the roots. Even in the case of Frankia, they are the group of mainly the actinomycetes. They make nodules in the woody plants, in the perennial plants in the temperate regions. They make nodules like tennis balls. So these are the other group of microorganisms which form nodules also. These are also type of biofertilizer classified on the basis of FCO specifications. So there are certain FCO standard is available. FCO means fertilizer control over order. They are listed in the fertilizer control over order, and there are specification has been given for the fertilizers, biofertilizers like for the rhizobium, Azotobacter, Azoospermum, phosphate solubilizing bacteria, and mycorrhiza. For these biofertilizers, a specification has been given by the fertilizer control order, and even these biofertilizers are listed in the FCO FCO Schedule 1. So that's why they can be marketed into the country. Other biofertilizer which are not covered under the FCO, that is the Acetobacter, Fertilizers aurantia, Pseudomonas, SPGTR, mixed cultures, and effective microorganisms. So these are not listed in biofertilizers, but they are sold in the market. Certain of them are in the market as PGPR, plant growth promoting bacteria, or with other trade names also. If you see the ecology of the nitrogen fixation, they can be classified into four groups. That is the system of nitrogen fixation, that in which the atmospheric nitrogen converted into the ammonia. And so the first one is the symbiosis, in which the rhizobium is the most important one. And in this case, root nodules are formed in the roots of the plants. And the source of energy which are required for them, that is sucrose, that is taken up by the host plants. So the photosynthesis process, they supply nutrients to these microbes for the energy, and by the nitrogen fixation, they supply nitrogen in the form of ammonia to the plant system. So estimation of fixation rate, nitrogen per hectare per year is 50 to 400. So quite a variation is there in the case of nitrogen fixation. That will depend upon what is the duration of crop. In the long duration crop, the nitrogen fixation is very high. But in short duration crop, like in mung, case of the where they are used uh, for the green burning process, 40 to 50 kg nitrogen fixation will be there. In the case of ammonia, same. One of the very high, it is up to 400 kg. In case of pigeon pea, it may be 100 to 120 kg. In the case of soybean, also it is 80 to 120 kg. Okay, so nitrogen fixation is very dependent upon the species, plants, and duration of the plants also. Then association, that in that case, azoospermillum will be there, is there, in which they are associated with the roots. They are attached to the roots. So they require less oxygen, less tension of oxygen. 0.02 atmospheric pressure is sufficient for the azoospermillum. So root exudates, where the exudation takes place, at that places these microbes are associated with the plants. They use the exudates as an energy source, and they multiply on the rhizoplane and fix atmospheric nitrogen, and about 15 to 20 kg of the nitrogen fixation will be there. तो 10 to 200 नहीं है, ये 10 to 20 है आपका. Then free living, rhizodo pyrolium is also there. So heterotroph plant residue also are in case of autotroph photosynthesis is used, and several free living bacteria are there that are able to fix atmospheric nitrogen, and 10 to 20 kg of nitrogen fixation is there. तो आपको अभी बताते हैं, सबसे ज़्यादा नाइट्रोजन फिक्सेशन राइजोबियम में होता है, 50 to 300 400 kg तक होता है. एक्टिनोराइजा जो कि है फ्रैंकिया स्पेसीज को कहते हैं, एक्टिनोमाइसिट्स के कारण, एक्टिनोमाइसिट्स इन राइजोस्फीयर एक्टिनोराइजा हुआ, फ्रैंकिया up to 150 kg nitrogen per hectare per year. Then azoospermillum is 10 to 20 kg, azotobacter is also 20 kg nitrogen per hectare. azoospermillum और azotobacter में क्या डिफरेंस है?
सर एजोटोबैक्टर नॉन सिम्बायोटिक और एजोस्पेरिलम एसोसिएटेड है.
और कोई डिफरेंस?
सर एजोस्पेरिलम नॉन लेगुमिनस क्रॉप में होता है एंड एजोटोबैक्टर सीरियल्स एंड वेजिटेबल्स में होता है.
दोनों जो कि है नॉन लेगुमिनस क्रॉप के लिए है, और ये एजियोस्पर्मम मेनली C4 प्लांट्स के लिए ज़्यादा इंपॉर्टेंट है एज कम्पेयर टू द एजिटोबैक्टर जो कि फ्री लिविंग है, और ये वाटरलॉग्ड एरिया के लिए नहीं है आपका एजिटोबैक्टर, जबकि एजियोस्पर्मम जो कि वाटरलॉग्ड एरिया में भी सर्वाइव कर जाता है. एजोला जो कि है 40 to 50 kg तक नाइट्रोजन फिक्स करता है. साइनोबैक्टीरिया अभी 20 to 40 kg तक. माइकोराइजा सोलुबलाइज्ड फास्फोरस दैट डिपेंड अपॉन द कंडीशंस और एनवायरमेंटल कंडीशंस आल्सो. Now, a scope of biofertilizer in agriculture. So, in the scope, if you see, they are able to supplement fertilizer supply for meeting the nutrient needs of crop. Supplement means क्या हुआ जी? सप्लीमेंट फर्टिलाइजर सप्लाई, इसका क्या मतलब हुआ?
सर
अल्टरनेटिव्स और कोई सप्लीमेंट का मीन्स अल्टरनेटिव केवल नहीं हुआ, दे कैन सप्लीमेंट, ओनली कैन सपोर्ट, है ना. आप टोटली यू कैन नॉट टोटली रिप्लेस द फर्टिलाइजर रिक्वायरमेंट ऑफ द क्रॉप बाय द एप्लीकेशन ऑफ बायोफर्टिलाइजर्स. Okay, so only you have to supplement them. Certain quantity of the fertilizers can be supplied, certain amount of nutrient can be supplied by biofertilizers, but you cannot cut the total amount of biofertilizers. But in case of legume, you can cut the nitrogen only. The supply of phosphorus and potassium will be required. Okay, so they cannot cannot replace the fertilizer. They only supplement. They only support the fertilizer requirement. They only you can deduct the dose of the fertilizers. Okay, so they can add 20 to 200 kg of nitrogen per hectare by fixation under optimum condition and solubilize or mobilize 50 to 50 kg of the phosphorus also. So besides nitrogen, they are capable of mobilizing or solubilizing the phosphorus also 50 to 50 kg. They liberate growth promoting substances and vitamins and help in plant growth. Besides nutrient supply through the fixation and solubilization or mobilizations, they are capable of promoting growth by secreting substances, synthesizing substances in the rhizosphere, which are supporting the plant growth by the plant growth hormones, by the vitamins also. Okay, so they supply these growth factors which are helpful for the growth of the plants also. In some cases, they suppress the incidence of pathogens and control diseases also. In the rhizosphere, like PGPR, they are, what are the mechanism of controlling the pathogens? Have you any idea? What is the mechanism for controlling the pathogens into the fields through the application of biofertilizers or PGPR?
Sir, they mop off certain element and not available for the other microorganism.
तो कैसे हुआ कार्बन सिक्वेस्ट्रेशन?
Yes, sir. Iron sequester, yes sir.
और एक और मैकेनिज्म बताए थे, CN वाला मैकेनिज्म बताए थे, हाइड्रोजन साइनाइड वाला, जिससे सिंथेसिस को रोक देता है. एक और है कि PGPR जो की है उसके लिए जगह ही नहीं छोड़ता है. ऑन द रूट सरफेस दे आर multiply or colonize in such a way there is no space for the pathogens. Okay, so there are several mechanisms, 4 or 5 clear-cut mechanisms through which they control the pathogen, they control the disease also. They increase crop yields by 10 to 50% nitrogen fixer, reduce depletion of soil nutrients, and provide sustainability to the farming system. In case of legume crop, they are able to supply the residual nitrogen into the fields. Okay, so that's why if you take a pulse crop into the field, in the next season the crop will be good, and due to the residual effect of the nitrogen into the plant system. So they increase the crop yield by 10 to 15%, and nitrogen fixer reduce depletion of soil nutrients and provide sustainability to the farming system. They improve soil physical property, health, and soil health in general, and they are cheaper, pollution free, and renewable energy sources. They are very cheap, very less cost are involved when you are applying to the fields. It is less than 100 rupees per hectare, or 100 or 200 rupees per hectare. So very less cheaper, and it is also pollution free, non-hazardous, and they are renewable energy sources. Now, these are the common mechanisms through which the nitrogen fixation takes place. Nitrogen fixation means the atmospheric nitrogen N2 is converted into the ammonia. Okay, so it is a rapid reduction process in the presence of the enzymes. So, there is nitrogen is converted into ammonia. So dinitrogens are reduced to the diamide, and that diamide is reduced to the hydrogens, and further it is reduced to the ammonia. So it is a continuous reduction process through which they are converted into ammonia in the presence of enzyme, and that enzyme is the nitrogenase. Okay, so this nitrogenase is capable of converting atmospheric nitrogen into the ammonia. So in this case, 6 hydrogen is required with 6 electrons, and then the 2 molecules of the ammonia is fixed. Okay, so the nitrogenase, which is the main components for the nitrogen fixation, if any organisms which have nitrogenase, they are capable of fixing atmospheric nitrogen. Otherwise, there is no nitrogen fixation process which takes place. So either Azotobacter, either Azoospermillum, either Rhizobium, either blue-green algae, either Azolla, each and every have nitrogenase enzymes. Okay, in the case of Actinomyza, also Frankia, they have also nitrogenase. Okay, so these nitrogenase are capable to reduce atmospheric nitrogen into the ammonia. Okay, so as you know also in the biochemistry you have learned also about the enzymes are mainly proteins. So these enzymes are also protein. Nitrogenase is also protein. It have two protein fractions. One is the molybdenum iron protein fractions, another one is the iron protein fraction. Okay, so the molybdenum iron protein fraction, it is also called molybdoferredoxin or azofermo. है ना, एजो मीन्स नाइट्रोजन, Fe फॉर आयरन एंड MO फॉर मोलिब्डेनम. इसको एजो फेर्मो भी बोलते हैं, है ना. आयरन प्रोटीन इट इज ऑल्सो कॉल्ड एजोफॉर. एजो मीन्स नाइट्रोजन, Fe फॉर द आयरन. ओके, देन इट इज ऑल्सो कॉल्ड एजोफेरोडॉक्सिन, है ना. नाइट्रोजन फिक्सिंग, जो नाइट्रोजन फिक्स करता है आयरन प्रोटीन, दैट इज एजोफेरोडॉक्सिन. Okay, so the nitrogenase is a protein and it had two fractions, that is molybdenum iron proteins and iron protein fraction. Molybdenum iron protein, its molecular weight is 220,000 to 270,000 and it is also called molybdoferredoxin or azofermo. The second portion is iron protein. It is low molecular weight materials that have 55,000 to 68,000 molecular weight is there, and it is also called azoferredoxins or azofer. Okay, so these nitrogenase, that is active nitrogenase, reconstituted by the addition of purified protein from the Bacillus polymeria or Klebsiella pneumoniae or blue-green algae. So in the laboratory, these nitrogenase can be reconstituted by the addition of the proteins also from the Bacillus polymeria, Klebsiella pneumonia, and blue-green algae. So these molybdenum iron proteins, that is the dinitrogenase nitrogen, bind to the protein molecules. Okay, so first at all, nitrogen is atmospheric nitrogen is attached with the molybdenum iron proteins. Okay, then it is attached to the iron protein, dinitrogen reducted to reduce molybdenum iron protein. So the main function of iron protein is to reduce them, okay, into the ammonia. Okay, so that in the enzymatic reduction process, the nitrogen, that is nitrogen, is converted into the ammonia to the two intermediate property diamide and hydrogens. So in this case, in the initial case, I have shown you that a 6 hydrogen is required, but here I have shown you that is 8 hydrogen is required, and 2 hydrogen comes as a product, byproduct. Okay, so here 8 hydrogens and 8 electrons when combined with the nitrogen, they are reduced to the 2 ammonia. Okay, so 2 ammonia with the 1 molecule of hydrogen. So hydrogen evolution is there along with the ammonia is inherent property of nitrogenase enzyme. So the nitrogen fixation through the nitrogenase enzymes, there is the release of hydrogen also, evolution of the hydrogen also. Okay, so in certain cases, these hydrogens are taken up by the plants again and it is recycled. Where the hydrogenase uptake enzyme is there, HUP positive, HUP positive enzyme is there, with which the evolved hydrogen is taken up by the plants, and in the certain plants there is no mechanism of the hydrogenase uptake. So in that case, that hydrogen is lost. Okay, so in case efficiency is lower, but in case where the HUP positive enzyme, HUP positive means hydrogenase uptake. Okay, hydrogenase uptake is present. So in that case, energy conservation is there. So less energy will be required when the HUP positive is there, and higher energy will be required when the HUP negative will be there. Okay, so in this case, 16 ATP energy will be required. Okay, so but the efficiency is very low. For that purpose, the requirement is about 35 ATP required, and 1 molecule glucose yield at maximum 38 ATP. Okay, so for synthesis of 2 molecule of ammonia, about 38 ATP will be required, and that will comes from the 1 molecule of the glucose. Okay, so 1 molecule glucose is equivalent to 2 molecules of the ammonia plus evolution of hydrogen molecules. Okay, so in the bottom I have given certain other compounds are also reduced and that are used in the laboratory for the estimation of nitrogenase enzyme also. Okay, like acetylene. Acetylene is also converted into the ethylene by the reduction process. Hydronium oxide is also converted into the hydrogen. In the same way, nitrogen is also converted N2O. Nitrogen is also converted into the ammonia. Okay, so cyanide is also converted into the methane. N2 is also converted into the N2. So all these are the reduction process, and in the laboratory they are used to measure the nitrogenase estimation, to the nitrogenase estimation, in which we use the gas liquid chromatography and a GLC, gas liquid chromatography technique, in which we put one sample of the acetylene. Okay, so acetylene are inserted into the container which contain nodules, and that acetylene when converted into ethylene. Okay, through the nitrification through the reaction process. So after certain period, we measure that gas whether the content of what is the content of ethylene. Okay, so that indicate the equivalent amount of the nitrogenase. Okay, so it is the nitrogenase used in the nitrogenase reduction assay. Now, mechanism of nitrogen fixation. So the in case of mechanism of nitrogen fixation, although it is mechanism is different in different types of plant systems and microbes also, but the main mechanism is the that it takes place through the nitrogenase enzymes. Nitrogenase reaction takes place in which the electron activator by its donor or ADP will be required. One electron activator will be there, that must be a pseudoner donor or ADP will be required. Okay, ADP is the energy currency, and that must be required during the process of nitrogen fixation. Then the second process, when the energy is available and a suitable donor is available is there, then the substrate reduction process takes place. Substrate reduction means the atmospheric nitrogen is converted into ammonia through the step by reduction process in which two intermediate compounds are formed. And it is, if you see the first process where the molybdenum iron protein, it is attached with the molybdenum iron protein, then it is attached with the, after that dinitrogen is goes with the attached with the iron protein, and then it is finally converted into the ammonia through the final reduction process. Okay, so in the substrate reduction, these processes, these two protein component of the nitrogenase, that is molybdenum iron protein and iron protein. So it takes place at different sites into the enzymes, that is 3.8 angstrom differ into the size on the sites and on the nitrogenase molecule, but are interdependent. They are interdependent because first nitrate substrate molecule are attached with the molybdenum iron proteins. Then it will impose to the for the reduction to the iron proteins. So the site difference is only 3.8 angstrom. So the nitrogenase, that is iron protein, is highly sensitive to oxygen. Okay, although protein is sensitive to oxygen, but the two components, molybdenum iron proteins and iron proteins, iron protein is highly sensitive to the oxygen. If oxygen is present there, they cannot work. They do not work for the reduction process. In the absence of oxygen or very less supply or less tension of oxygen will be required. Okay, so iron protein is really highly sensitive to the reductions through the oxidation process than the molybdenum iron protein. So in the microbes, it is believed that an unidentified respiratory system exists. Okay, so in different type of microbes, like in rhizobium, there are different mechanism. Azotobacter, there are different mechanism for the saving of nitrogenase. In the azoospermillum, there are different mechanism for the saving of nitrogenase. In the case of blue-green algae, there is also different mechanisms. In case of azolla, there is also different mechanism. So each and every microbes, there are different mechanism to conserve nitrogenase, because that is the main molecule, most important molecules that is responsible for nitrogen fixation. They are sensitive to oxygen. So they have to make away from the oxygen. And for this purpose, there are several type of mechanism are there. So in the microbes, like in Azotobacter, if you see, there it is believed that an unidentified respiratory system exists in the Azotobacter. As you know, Azotobacter is free living. They require more oxygen for respirations. Okay, so near the site of nitrogen fixation, where the nitrogenase enzyme is there. Okay, so they actively convey and prevent inactivation of nitrogenase. So they respire very fast, and in that way, they uh, conserve the nitrogenase enzyme by scavenging the oxygen at the site of the nitrogenase enzyme. Okay, so this technique, usually this type of mechanisms are available with the Azotobacter. That's why due to the high respiration rates of the Azotobacter, they require more carbon materials for their food materials. That's why Azotobacters fix atmospheric nitrogen more in the organic matter rich soil. If organic matter content in the soil is very less, then the food requirement of Azotobacter will not met by the for the respiration. Because they respire fast, so they oxidize organic matter very fast. But when the organic matter is not present in sufficient amount, their multiplication will be restricted, their respiration is restricted, and the nitrogen is not conserved. They are not able to conserve their nitrogen, and that's why the nitrogen fixation is very low in that cases. Okay. Now, this is the one of the mechanisms through which the nitrogen fixation takes place. So, in the system, there is the through the pyruvates, there is the phosphatoclastic reactions will takes place, through which the acetyl phosphate and the ADP are generated simultaneously, and through the acetyl phosphate and the ADP, they are converted into ATP that will be act as a energy for the electron donor for the nitrogenase enzyme. Okay, so in the nitrogenase enzyme, both type of molybdenum iron protein and iron proteins are there. Okay, so nitrogen will be attached with the molybdenum iron proteins, and in the presence of the energy ATP, they are they start reductions of the nitrogens into the different reduced molecules through the molybdenum iron proteins, and then through the iron proteins they are reduced to the ammonia. Okay, so here from the pyruvate there will be release of two hydrogens. That hydrogen will be utilized to the heterodoxins for the reduction of nitrogen into the ammonia, and here the evolution of the hydrogen will be takes place. So these are the simple mechanisms through which the nitrogen fixation takes place. Okay, like once again I explain you that is the through the pyruvates there will be simultaneous through the phosphatoplastic reaction, simultaneous reaction of acetyl phosphate and ADP will be there, and there will be synthesis of ATP that will work as energy for the nitrogenase, and nitrogen attached with the molybdenum iron proteins, and that will be the ammonia through the hydrogens which are released to the pyruvate that will be attached to the ferrodoxins, and they will be the ferrodoxins that will finally release the nitrogen into the ammonia by the evolution of the one molecule of hydrogens. Okay, there are several other processes in the laboratory can be used. That is the lithionite is also there. That is iron sulfur electron carrier protein can also be used for the reaction also. Reversible oxidation reduction strong is there, and a strongly reducing electron carrier protein is there. So the thionate in the laboratory is also used. Another one is the flavotoxin, that is in the case of Clostridium fasterinum and disulfur vibrio species, azitoflavin in case of Azotobacter vanillandi, they can be also used as like a ferrotoxins. reductions in the Azotobacter in Azospectrum is also there, but in certain cases, like in the Clostridium, there is a flavotoxins. In case of the Azotobacter, Velanandi, there is a azotoflavin is also there for the reduction of nitrogen. Some dyes are also used for the reduction process. In the case of methyl halogens and benzyl halogens are also used for the reduction process. They are strong reducing agents. They can be used in the laboratory to demonstrate the nitrogen fixations. Okay. Now the mechanism to prevent nitrogenase inactivation. So I have discussed you that there is different mechanisms for conservation of nitrogenase, because that is the key element for the nitrogen fixation. If nitrogenase is active into the microorganisms, they continuously fix atmospheric nitrogen. If they are not active, they are in an inactive stage, the nitrogen fixation will be affected. Okay, so there are different mechanisms. So the first mechanism is the respiratory protection of nitrogenase, as we discussed in the case of Azotobacter. Azotobacter is aerobic in nature. They require organic matter for the food. So, and they save their nitrogenase by the fast respiration process. Through the fast respiration process, they scavenge the oxygen from the nitrogenase surface. Okay, so in the microbes, it is believed that an unidentified respiratory system exists in Azotobacter near the site of nitrogen fixation, which actively scavenges and prevent inactivation of nitrogenase. In case of cyanobacteria, also this type of process is there. Second one, and second one is the conformational protection. Some conformational protection is there, that is developed in nitrogenase that changes to a protective conformation in the presence of oxygen. So in the presence of oxygen, they are inactivated. Some protective layer will be developed. So in the protected state, fixation is totally reloaded. But the activity rapidly returns when the oxygen tension reduced. So there is some mechanism. So in the presence of oxygen, protective conformation will be there, and in that case, the nitrogenase is become inactive. But when the fixation is totally depleted, then the activity rapidly returns when the oxygen tension is reduced. So when the oxygen is removed from the surface or it is reduced, then the activity will be rapidly. Third one is the oxygen regulation by nitrogenase synthesis, prevent nitrogenase production under unfavorable conditions. So in certain cases, there is a oxygen regulations by the nitrogenase synthesis, that is prevent nitrogenase production, prevent the production of nitrogenase under the unfavorable conditions, or when the oxygen level is very high, then the nitrogenase production is prevented, like in the case of Klebsiella pneumonia and Azotobacter coccopo. So in these cases, certain cases, it was found that there are certain mechanisms through which, in the presence of oxygen, they did not synthesize the nitrogenase. But in the very low tension of oxygen, the synthesis of oxygen will take place. Another one is the mechanism is the gum production. That is, the nitrogenase is protected by physical barrier to molecular oxygen. So certain microbes produce gums or polysaccharides that accumulates over their external cell wall, and that are the barrier for the oxygens. Only reduced amount of oxygen for the respiration are there, like in the case of Tertia gummosa, where the cell on the cell wall, a huge gum layer or polysaccharides are accumulated that prevent them to supply oxygen, and they are capable to fixed atmospheric nitrogen, and they save the nitrogen. Even in certain azotobacter, this type of mechanisms are also there. Okay. Another one is the head to cyst production. Like in the case of blue-green algae, there are heterocyst formation are there. Heterocyst formations in which the vegetative cell walls are converted into the thick layer. Multiple cell walls are there, and the thick layer will be developed. And under the thick layer conditions, the diffusion of oxygen is very low. Okay, so heterocyst production in which the molecular oxygen is the product of carbon fixation in aerobic conditions, nitrogen fixer, cyanobacteria. In these, nitrogenase enzyme is isolated in a structure with thick wall cell to reduce oxygen contact. Okay, so in the several blue-green algae, uh, some vegetative cells are converted into the heterocyst. Heterocyst means the cell walls have multiple layers of cell walls will be there. The cell walls of the cells, vegetative cells, have multiple layers, and that why they are very thick. And due to thickness of the cell walls, they reduce the oxygen supply, or movement or diffusion of oxygen through the cell walls are restricted. And in that case, nitrogenase remain in active stage or remain protected for the nitrogen fixation. So in the blue-green algae, there are series of vegetative cells, and between them there may be one or two heterocyst cells are there. So the heterocyst, in the heterocyst cell, nitrogen fixation takes place, and in the vegetative cells, photosynthesis will be takes place, and six carbon compounds or polysaccharides or sugar compounds are synthesized. They are supplied to the heterocyst for the nitrogen fixation. Okay, and after fixation, these nitrogens are converted into ammonia, and their ammonia is supplied to the vegetative cell for the growth. Okay, so heterocyst production is also one of the mechanisms through which the oxygen diffusion through the atmosphere to the plant cells or through the heterocyst cells are restricted, and they conserve the nitrogenase enzyme into the heterocyst cell for the further nitrogen fixation. Okay. Sixth one is the mechanism of leg hemoglobin production. So in the case of the symbiotic nitrogen fixation in the rhizobium, in case of rhizobium, if you see the leg hemoglobin is there. As in the human body, blood containing hemoglobins. That is the hemoglobins. What is the function of hemoglobin? They supply oxygen to the body, different parts of the body. Okay. In the same way, in the rhizobium also, in the legume plants, leg hemoglobin is there, and their main function is to supply oxygen at reduced tension into the nodules. Nodules are mainly that contain leg hemoglobin, and rhizobium is imbibed into the leg hemoglobin, and these leg hemoglobin are synthesized in the cytosol. Okay, so through the cytocells, after synthesis, they are transferred to the nodules, and nodules is full of leg hemoglobin. It is pink in color, and if you uproot a legume plants with the having root nodules, the pink color of the nodules is identified as a active nitrogen fixing nodules. Okay, so these when rhizobium is imbibed into the leg hemoglobin, the nitrogen fixation rate is very high. Okay, because they control the oxygen supply. So in rhizobium, the nodules molecular oxygen is complexed with the leg hemoglobin and synthesized in cytosol and cause reduction of 0 up to 10 nanomole of the oxygen will be there. Reduction of oxygen up to 10 nanomole will be there. Okay, so under reduced condition, oxygen supply will be there to the leg hemoglobin to the rhizobia. Another one is the migration to suitable environment. So the in there is symbiotic nitrogen fixation. They move to the plant system as endophytes and make nodules. But certain microbes which are remaining to the soils, so they migrate to the suitable environment. Okay, so aerotactic organisms are there. These organisms which migrate to the suitable environment for nitrogen fixation are called aerotactic microorganisms, and they may migrate to position in their microenvironment that is suitable for nitrogen fixation. Like in the case of azoospermia, they may migrate in the soil or uh, in the rhizoplane, on the rhizoplane of the roots, through which they can protect their nitrogenase enzyme. Okay, that's why there is a micro association will be there with the plant roots. Okay, so these are the mechanisms to keep the nitrogenase is prevented from the inactivation. Okay, thank you for today. अभी क्लास है ये audible?
Yes, sir.
अभी क्लास है या खाली हो तुम? बोलो क्या हुआ जी? ज्योति
यस सर।
अभी क्लास है या खाली हो?
सर अभी तो
फ्री है तो क्लास कंटिन्यू रखें, आधा घंटा और बता देते हैं, थोड़ा सा और है तो बहुत सारा, लेकिन आधा घंटा और देख लेते हैं, ठीक है?
सर एक बार बाकी मतलब बच्चों से भी पूछ लीजिए, बाकी तो ठीक है।
मैं तो पूछ ही रहा हूँ, तुम लोग कुछ बोल ही नहीं रहे हो। हाफ एन आवर और कंटिन्यू रखें जी। बोलो।
यस सर।
चलो ठीक है। ये this table shows estimation of biological nitrogen fixation on global basis. So if you see from the legume, it is about 35 million metric tons nitrogen fixation annually takes place throughout the globe. Okay. In the case of non-legumes, it is about 9 million metric tons. For permanent grasslands, it is 45 million tons. From the forest and woodland, it is about 40 million tons. And from the unused land, about 10 million tons. And from the oceans and sea, it is about 36 million tons. So total nitrogen fixation globally that takes place is about 175 million tons. So a huge quantity of the nitrogen fixation takes place on the Earth's surface through the beneficiary microorganisms. And the industrial nitrogen fixation is far less than the biological nitrogen fixation. It is less than 100 million tons per year. Okay. Now there are classifications of biofertilizers, and that may be classification on the basis of some of the biofertilizers are formulated on the basis of with the carrier materials, and some of them are formulated without carrier materials. So two classification is there. One biofertilizers are those which are manufactured with the carrier materials. Another one is without carrier materials. Okay. So the carrier-based inoculants containing cells of efficient strains of a specific microorganisms, mainly bacteria, used to enhance the productivity of the soil, either by fixing atmospheric nitrogen or by stabilizing soil phosphorus or by stimulating plant growth through synthesis of growth promoting substances. Okay, so in this category, another category is the blue-green algae or cyanobacteria, in which the soil flags are used. Then the another category is the azolla that can be used also for the nitrogen fixation. The green azolla is used as seed materials. third group is the mycorrhizae, in which the mycorrhizal association roots are mainly cut down and they are supplied to the market with the vermicompost or compost materials. So 3 or 4 groups are there. Mainly that one is the carrier-based materials, in which all the bacterial culture are there. Second one is the blue-green algae or cyanobacteria. Third one is the azolla, and fourth one is the mycorrhizae there. Now, in the carrier-based biofertilizers, all bacteria are considered under this group, in which the rhizobium is one of them, Azotobacter, Azoospermillum, uh, and other the phosphate solubilizing bacteria and potassium solubilizing microorganisms are also considered under this category. Okay, so in the case of rhizobium, if you see rhizobium, Azoospermillum, Azotobacter, Acetobacter, phosphate solubilizing bacteria, so all the bacteria are used for the manufacturing of biofertilizers and carrier-based materials. Okay. So in case of rhizobium, uh, belongs to Rhizobiaceae family, and Azorhizobium for stem nodulation is there in the case of Sesquenia rosata, and another group important, although there are 5 groups, but here we are discussing about the Azorhizobium, that is for the stem nodulations, Bradyrhizobiums for the slow growth takes place in case of Bradyrhizobium, in case of spam, copy, miscellaneous, copy, gram, and ground, all those cases that is the past in the case of okay. So the morphology of these bacteria that are essentially less than 5 microns, and just from being broad right there. You know, motile and ground is negative. Okay, so these are the descriptions and characteristics of rhizobium. Okay. The physiology of rhizobium are there heterotrophs, sensitive to their limbs also, and are also satisfied by the limbs of the substrates and anti-monocytes substrates also. Nitrogen also satellite from the atmosphere of space nitrogen used, and the applications that are applied by plant direct. There is no chemical development there, and that's why there is residual effect also. That's why that's why but the function they become some other than nitrogen requirement in the oxygen requirement in very. That's why they are growth that is far that and slow growth they produce alkali into their surroundings in the microbiome. So growth is fast for the rhizobium and slow growth of rhizobium. In the case of rhizobium, they produce acid with the multiplication, and in this case they produce alkali with the multiplication. Doubling the type of hard growth that is the rhizobium to 4 hours, for the slow growth to microbiome it is 6 to 12 hours. Okay. Growth here there are certain oscillations on the basis of CCU, and they are 5 to 7. Okay. Those oscillations are made to identify the hot plant in which this type of microorganisms are associated. So the group of organisms are associated with the hot plant. If you locate that plant, then there will be no association there. If these are multiplied by another plants, another legumes, then the nitrogen fixations are affected. Okay, like in the case of rhizobium, rhizobium vacea, that is for the vacea host legume is there. Rhizobium leguminosaurum, it is trifoliate valually and trifoliate valuelus. Rhizobium melureti is there, rhizobium fridi is also there. In the case of bradyrhizobium, that is slow grower species, loti is there for bradyrhizobium japonicum is there for the glycine max. Bradyrhizobium species is there for the miscellaneous group. that is Vigna, Kaisner, Caesar, Lupins, all these things. So these are the cross inoculation plants are there. That is important for the inoculations. That's why in the case of inoculation, when you are going for inoculations, if the crop name is given, that is rhizobium can be used for this crop, that means that only can be used for that crop only. Contribution, if you see the contribution of the rhizobium, that is the highly variable, and if you see the box are capable of fixing nitrogen very high, that is 100 to 200 kg, followed by the PGP 168 to 200 kg, clover 100 to 150 kg, chickpea 85 to 110 kg, cowpea 80 to 85 kg. So several legume plants are there. Groundnut 50 to 60 kg, lentil 90 to 100 kg, green gram 50 to 60 kg, pea 52 to 77 kg, soybean 60 to 80 kg. So they are contribution of the nitrogen symbiotically with the legume plants. Besides this, these are the direct contribution. Besides this, there is a residual nitrogen benefit for the subsequent crop. Okay, so they contribute 30 to 40% of the nitrogen to the next crop also. So then after harvest of the crop, second crop is taken, they get benefited from the residual nitrogen of 30 to 40% of the free nitrogen. Okay, recommended for the pulse also, like pea, lentil, black gram, green gram, cowpea, BMB. Then oil seeds like soybean, groundnut. For the fodder, berseem, Egyptian clover, dosal. For the spices like methi and green mangoes are important for the recommendations, and there a huge amount of nitrogen fixation will be there. There are chances of increase in yield up to 10 to 35% besides the another one is the Azotobacter, that is aerobic in nature, and the family is the Azotobacteraceae, and the species are Azotobacter prococum, that is identified or isolated by Rinki in 1901. They are suitable for the neutral and alkali soil, and very crackers, flag lie also there. In the same way, Azotobacter that is isolated by the Lippmann in 1903 in the north commercial soil. They have also peritrachus, flag lie there. The Azotobacter is there. Isolated by the in 1904 from the north commercial soil, and they have no flag liability there. The Azotobacillus nigricans, that is Nikov in 1949, and the Azotobacter phosphate, that is the Duvernier in 1960 from the Egyptian soils, which have also peritrachus fungus flag lie there. Another one is the Azotobacter agilis, that is now categorized under the Pseudomonas, and Azotobacter insignis, that is isolated in Indonesia, have also peritrachus flagella also. Azotobacter ecocystogens, they have also peritrachus flagella, and they are aquatic in nature, and they are isolated by the in 1938. Okay, so these 6 group of Azotobacters are important for the different regions. In the India, we are using only the Azotobacter prococum. They are abundant in soil. They can be isolated from anywhere from the rhizosphere of the soil. Okay. So morphology, cell size is large, ovoid cells, size ranging from 2 to 7 micron in length and 1 to 1.5 micron in diameters. Cell characteristics, they are polymorphic, accumulate, poly-B granules, and gram reaction is negative. तो physiology जो की है, that is chemoheterotraphic is there, free living, carbon source, variety of carbon source is required, mono, di, or certain polysaccharides. There are organic acids are also required. That is the atmospheric nitrogen can be used, even amino acids can be used also, ammonia is also used, nitrate is also used. Respiration, that is strictly aerobic, and growth, that is growth media will be absent media or Johnson media is will be used. doubling time is about 3 hours. So the benefit of these introvector, that is that introduce of 20 to 40 milligram of the beneficial nitrogen fixation will take place. Biological nitrogen fixation will take place. Carbon source in laboratory conditions, that is equivalent to about 20 to 40 kg nitrogen per hectare. Okay. Now production of growth promoting substances like vitamins, B groups, indole acetic acid,
उसके साथ निकल गया।
आ रहा है प्लस में। Now biological control by suppressing Aspergillus fusarium. They can control plant disease also by suppressing Aspergillus fusarium. Then recommended for rice, wheat, millets, other cereals, cotton, vegetables, sunflower, potatoes. Okay, so they can be recommended for the rice for the upland soil only, and wheat, millets, other cereals, cotton, vegetables, sunflower, mustards, ornamental plants, for each and every we can recommend that. Another group is the Azoospermum, that is family Asparaceae, and it belongs to the family Asparaceae, and number of species are 4, that is Azoospermum lithopterum, Azoospermum brasiliensis, Azoospermum amazonix, and Azoospermum hello preference or seropetici. But in Indian conditions, Azoospermum brasiliensis are mainly prominent in the fields. The nature is mainly chemotrophic, they are associative, mainly they are taken organic acids or arabinoids or gluconates, and deep fructose, deep glucose, sucrose, pectin, variety of carbon source they can utilize. Nitrogen source, that is nitrogen for fixation, acids, ammonia, nitrate, they can also take. So for the media formulation, media growth, they can be used. Respiration, aerobic or microaerobic in nature, growth media, bromothiamine, blue medium, et al. 1975 is used. Then doubling time is 1 hour on a ammonia containing medium and 5.5 to 7 hours on a millate containing semi-solid media. Okay. ऐसा क्यों है जी, doubling time इतना variation क्यों है? Why there is a variation in the doubling time 1 hour on a ammonia containing medium and 5.5 to 7 hours containing semi-solid medium? बताओ कोई? ज्योति, कहाँ गया सब्जी? मैलेट मीडियम में क्यों इतना ज्यादा समय लग रहा है जी ग्रोथ में, और अमोनिया कंटेनिंग मीडियम में 1 ही घंटा लग रहा है?
अमोनिया कंटेनिंग मीडियम में डायरेक्ट सोर्स ऑफ
क्या चीज? अभिनव फिर से बोलो।
सर अमोनिया में अमोनिया वाले मीडियम में डायरेक्ट सोर्स ऑफ नाइट्रोजन अवेलेबल है। ग्रोथ फास्ट होता है।
मैलेट मीडियम नाइट्रोजन फिक्सेशन खुद करना पड़ता है ना, तो इसलिए सिंथेसाइज उसको करना पड़ेगा नाइट्रोजन अमोनिया का फॉर्म में। चलो ठीक है, आज इतना ही रहने देते हैं, ठीक ना, अगले क्लास में फिर देखेंगे।